Okay, so we're welcome to province-wide pathology rounds. Before we introduce today's speaker, I'd just like to note that next week, Professor Lucy Peroni is going to be giving a presentation as part of the PALS lecture series focused on quality assurance, and we look forward to that. Today, we have Dr. Kevin Kuchinski, who's going to be presenting on avian influenza. And Kevin completed his PhD in pathology and laboratory medicine at UBC, and he works in the BC Centre for Disease Control. And he's working on the laboratory side of developing tools for controlling and studying viruses in difficult specimens, including those, of course, which he has to work with when trying to control avian influenza. This is an area which I need to know much more about, so I really personally welcome the chance of learning from you today. And we'll have the presentation, and then there should be some time for questions afterwards. And so, Kevin, thank you for joining us today and presenting. And we look forward to an interaction with you after your talk.
Great. Thanks for the introduction, Dr. Huntsman. Thanks to all of you for spending your lunch hour at pathology rounds as well. Today, I'm going to tell you about avian influenza, and I'm going to tell you about how we're sequencing it in mud of all places for the purposes of surveillance. So I'm going to begin by introducing the influenza A virus. I think the most important thing to know about it for the purposes of today's lecture is that it's an extremely diverse virus. In fact, it's so diverse that it needs to be subtyped. So this subtyping is based on two hypervariable surface proteins called hemagglutinin and neuraminidase, or HA and NA for short. So when you hear about H1N1 or H3N2 or H5N1 or H7N9, those are all just different subtypes of the flu A virus. There's 16 HA subtypes, and there's 9 NA subtypes, and then each of those subtypes can be further subdivided into numerous different lineages and clades and variants. And these subtypes, they often become shorthand for referring to specific strains and lineages, like how we call the human seasonal flus H1N1 and H3N2. But it's really important to keep in mind that those subtypes aren't actually unique identifiers. They're very broad categories. So human seasonal H1N1, for instance, it's very far from being the only H1N1 influenza virus out there. Now, this extreme genetic diversification of flu viruses is really driven by two properties of the flu genome. First of all, it's an RNA genome, so it mutates quite easily compared to DNA in a process that we call genetic drift. And it's also a segmented genome. So the genome's actually split across 8 different pieces of RNA. And then when multiple flu viruses infect the same host, those different pieces of genome, they can get shuffled together, which creates new combinations in a process that we call reassortment. So because of genetic drift and because of reassortment, there's constantly new lineages and potentially new threats of flu emerging. Almost all flu lineages emerge and circulate in wild birds, especially waterfowl and shorebirds. So in other words, waterfowl and shorebirds are the natural reservoirs and hosts of flu A viruses. And since many wild birds are migratory, flu A lineages are able to disperse globally along migratory routes. Now, as humans, we really start to care about flu viruses when they spill over from wild birds into our poultry. High pathogenicity strains cause really severe disease in poultry. Outbreaks can wipe out an entire flock, leading to farm closures and quarantines and trade restrictions. So these outbreaks are very, very costly. But sometimes these outbreaks also continue spilling over into humans, which causes really severe novel influenza infections, which are popularly known as bird flu. These novel flus tend to have very high case fatality rates, anywhere from 30 to 50%. And there's been several notable examples in the past couple decades, like H5N1, H7N9, H5N6, and a smattering of other lesser known subtypes. Now, these novel flu infections are also a major pandemic threat. First of all, there's the risk that these viruses directly adapt to humans in these human bird flu cases. But there's a second larger risk, which is that these avian viruses also spill over into swine. Now, swine physiology allows them to be readily infected by both avian and mammal adapted lineages, which creates opportunities for that genome reassortment phenomena, which combines mammal adaptation with antigenically novel, highly pathogenic avian segments. And this has actually been the process by which all human flu pandemics in the past century began. I've listed the 4 pandemics on this slide. There was the 1918 H1N1 Spanish flu, the 1957 H2N2 flu, the 1968 H3N2 flu, and the 2009 H1N1 swine flu. Now, eventually, all of these pandemic influenzas become endemic, and they can linger for decades, causing the seasonal epidemics that we call seasonal flu. And all seasonal flu today is descendants of either the 2009 H1N1 swine flu or the 1968 H3N2 swine flu. Now, seasonal flu remains among the 10 leading causes of death worldwide, and it's very likely that this burden is significantly underestimated. So I just want to take a quick moment to tackle some misconceptions that exist about human flu versus bird flu. I think most people imagine flu as fundamentally a human respiratory pathogen, so they consider bird flu as something that's kind of rare and exotic, when in fact the complete opposite is true. Influenza A is fundamentally an avian virus, and the handful of strains that circulate in humans are actually the rarity and the exception. Like, out of the thousands and thousands of strains that exist of this virus, there's only been 4 of them that have achieved widespread circulation in humans in the last century. And I think another misconception is that human flu and bird flu are separate, largely unrelated phenomena, when in reality, they're all the same virus. So all human flus were once bird flus. And not even all that long ago, when you think about our history as a species, it's only been a few decades. So I suppose if we really wanted to be provocative, we could even say that each and every one of us has had bird flu at least a few times in our lives. And like the majority of human pathogens, they entered humanity through our food system, which is an enormous, constantly expanding exposure interface between us and animals. So really, I think we ignore these connections between bird flus and human flus at our own peril. So hopefully at this point, it's clear that flu viruses have major impacts on human health and human livelihoods. And it should also be clear that all these impacts start when wild bird viruses spill over into poultry. So if we can prevent these spillovers, we can avoid these impacts. And this is exactly the purpose of flu surveillance in wild birds. And it's why flu surveillance in wild birds is a cornerstone of outbreak prevention and pandemic preparedness programs in jurisdictions across the planet. So there's 2 main approaches to wild bird surveillance. The first is called active surveillance. That's where you go out and you capture live birds and then you test them for flu. And the second approach is called passive surveillance. And this is where you opportunistically sample dead animals. Now, some of these animals are found and reported by citizens. Others are voluntarily submitted by wildlife rehabs and hunters. But both of these approaches have major limitations and biases. So in the case of active surveillance, it's really labor intensive. The detection rates are very low, and there's a bias towards abundant species and species that are easily captured. Now, when you look at passive surveillance, well, you're only really sampling unhealthy animals or species that die from their flu infections, which means that you don't sample healthy asymptomatic carriers who are usually the reservoir species and the ones responsible for spreading the flu virus along the migration routes. And then in the case of hunter-killed animals, you have a bias towards species that are legal and popular as game, and then you're only getting your collections during the the official hunting season. And because of all of these biases and limitations, bird surveillance almost never detects incursions of dangerous avian flus before outbreaks start happening. So due to these limitations, our team has spent the last several years developing a new approach for bird flu surveillance. And this approach is based on testing environmental specimens from wild bird habitats instead of testing the birds themselves. So wild birds shed flu viruses in their feces and their mucus, and all this excreta accumulates in the sediment of their wetland habitats. So we figured that the sediment is like an archive of locally circulating flu viruses from diverse hosts, including rare and cautious avian species that normally escape our surveillance programs. And then on top of that, these specimens have the advantage of being very easy to collect, because as you can probably imagine, it's a lot easier to scoop some mud into a specimen cup than it is to go tackle an angry goose and shove a swab inside his hind parts. So now our ability to use these specimens, though, it really hinges on our ability to detect and characterize viruses in them. So basically, we need to be able to sequence flu genomes from these specimens. But environmental genomics for viral surveillance faces a couple of major technical challenges. So the first challenge is that the target genomic material is extremely dilute in these specimens. The sediment contains huge, complex microbial communities, and it's also largely composed of decaying tissues from plants and animals, which have comparatively enormous genomes. So this makes environmental genomic surveillance basically like looking for needles in a haystack. Another major challenge is that the viral genomes are fragmented. So this happens in the environment through a number of different biogeochemical processes, but it also happens in the laboratory during our processing of the specimens. And this fragmentation is really a problem because we expect environmental specimens to contain multiple viruses from multiple infected hosts. And we need to be sure that we can analyze these fragments without accidentally putting them together incorrectly and creating artificial fictitious genomes that didn't actually exist in the specimen. So this may be a bit of a forced analogy, but it's like trying to put together jigsaw puzzle pieces from multiple puzzles that have been mixed together, except nobody can tell you how many puzzles there's supposed to be, if any of those puzzles have all their pieces there, or what any of the puzzles are supposed to look like. So to address these challenges, we developed a novel platform for sequencing flu viruses in sediment. I'm going to briefly walk you through what happens. First, we extract RNA from 2 grams of sediment, which is about a teaspoon of mud. Next, we build sequencing libraries from that RNA. Then we capture and amplify the fragments of flu genome in those libraries using custom hybridization probes. And then we sequence all of the captured fragments. We also have developed custom bioinformatic tools to analyze and interpret the raw sequencing data. There's a tool that will actually resolve each separate distinct fragment of genome that was captured. And then we have other tools that subtype and further characterize these fragments, which is what I'm going to show you in a few minutes. But first, I'm going to point out a couple things that really set this platform apart from standard sequencing methods that you might be familiar with. So first of all, for the target enrichment, we do that by hybridization probe instead of multiplex PCR. I've got a little cartoon here illustrating the basic probe capture workflow, but I'm not going to go through it step by step, because I think really the most important thing to know about probes is that they're significantly less impacted by genetic mismatches than PCR primers. So this technique is way more effective at enriching diverse and hypervariable genetic targets like flu viruses. For this surveillance project, we created a custom panel of 9,370 probes. This panel targets all segments of the flu genome, and it targets all subtypes that circulate in birds, swine, and human hosts. Now, another thing that sets this platform apart from the conventional methods is that it can actually resolve distinct fragments of genome that have been captured. This is done using a specialized lab method called unique molecular indexing. So most standard sequencing methods, they generate a single consensus sequence for the specimen. Now, since our specimens contain mixtures of viruses from many hosts, getting a single consensus sequence doesn't really work for us. So our method uses these unique molecular indexes to generate basically a separate sequence for each fragment of genome in the specimen, which means that we can count and characterize each fragment separately. And it also gives us really highly accurate removal of sequencing artifacts and correction of sequencing errors. Now, before we get to the results, I just want to take a couple minutes to introduce the goose guangdong lineage of H5 viruses, because we're going to talk a lot about them today. They are currently public enemy number 1, and they are the global surveillance priority. This lineage emerged in farmed geese in Guangdong province, China in 1996. It caused the original H5N1 bird flu outbreaks in Hong Kong and Southeast Asia in the late 90s and early 2000s. And over the past 25 years, this lineage has been responsible for literally thousands of outbreaks in poultry and humans. And since it's been circulating around for so long, it's diversified into numerous different clades. Now, in the past decade, there's one particular clade of goose guangdong that has been responsible for most H5 outbreaks. It's called clade 2344. There's 2 recent H5 outbreaks in British Columbia that were caused by clade 2344 viruses. There was an H5N2 outbreak in 2014 and 2015, which actually grew into a North America-wide epidemic. And then there's the ongoing H5N1 outbreak, which is part of a global epidemic originating in Europe and Asia. I just looked up some current stats about the H5N1 situation in British Columbia. At the moment, there's 19 active farm outbreaks. Since April last year, there's been 104 affected farms. And since April last year, 3.7 million poultry have died from H5N1. And remember, that's just for British Columbia. So obviously the numbers get much bigger if you factor in the rest of Canada and countries from all over the world. And then finally, this clade 2344 isn't only concerning because it's having a huge impact on the poultry sector. It's also increasingly causing high mortality in wild birds. And it's also spilling over into wild mammals that eat birds, like foxes, skunks, and sea lions. It's also spilling over into domestic mammals like mink, which is especially concerning because mustelids have very similar respiratory physiology to humans. In fact, ferrets are the model animal for lab studies on influenza. And then finally, there's also been isolated human cases of clade 2344. So now you can probably understand why clade 2344 goose guangdong H5 viruses became a surveillance priority for public health and agriculture agencies in British Columbia. And since the fall of 2021, we have collaborated with the BC Ministry of Agriculture to conduct bird flu surveillance using wetland sediment and our probe capture sequencing technology. So the data I'm going to show you today comes from the past 2 surveillance seasons. So basically last fall, winter, and then the fall, winter before that. The avian flu surveillance season overlaps with wild bird migration season. So all of these collections occur between October and January. And over these 2 seasons, we collected 840 sediment specimens from 28 different wetlands around the Lower Mainland, the Fraser Valley, the Okanagan, and the Whistler area. And all of these locations are indicated on these maps by little red dots. And if you look at the second map, which is just the Lower Mainland and Fraser Valley, you can see that there's a nice mixture of urban wetlands like parks, there's semi-urban wetlands, and then there's rural wetlands as well. So to identify specimens that are actually worth sequencing, we screen everything by qPCR. So we detected flu viruses in 187 sediment specimens, which is a 22% detection rate. We had capacity to sequence 164 of the positives. And this box plot here shows you the qPCR CT values of the specimens that we sequenced. So these CT values, they ranged from 28 to 45, and the median was 37. So if you know anything about CT values, this should be telling you that flu viruses were barely detectable in these specimens. Like there was probably only 1s to 10s of genome copies in the specimen. So really needles in a haystack here. Nonetheless, though, we recovered almost 19,000 fragments of flu genome from these specimens. And this bar plot here shows that we were recovering thousands of fragments from each segment of the flu genome. Now this bar plot shows the sensitivity of the probe capture sequencing platform we developed. So flu genome fragments were recovered from 94% of the specimens we sequenced, although the sensitivity for the individual segments ranged from 69% to 78% because not all specimens had all segments present. But when you think about how weak the detections were by qPCR, these results show that the sequencing platform is still pretty effective at finding the needles in a haystack. Now, this bar plot here, it shows the number of wetlands in which we detected each HA and NA subtype. And basically it shows that we detected a lot of flu subtype diversity. We detected 12 of the 16 HA subtypes, and we detected all 9 of the NA subtypes. So these results really supported that notion that the sediment is this archive of diverse, locally circulating influenza viruses, and a great place to go for surveillance. We also looked at diversity of flu viruses within each specimen, and surprisingly, we found that 38% of the specimens had more than one HA subtype, and 35% of the specimens had more than one NA subtype. And there were actually even specimens that had up to 8 different HA or NA subtypes in them. So this data, it told us that we frequently detect multiple viruses in a single specimen, which really highlights the value of this sediment as a surveillance specimen, because you can get and see a lot of viral diversity just in 1 teaspoon of mud. Now, these results also did highlight that the flu genomes are very fragmented. The median fragment is only 223 nucleotides long. For your reference, the flu genome segments, they range in length from 900 to 2300 nucleotides. So basically, the median fragment only covered about 14% of its genome segment. Okay, so for surveillance purposes, we generally want to know more about the virus than just its subtype label. We want to know what kind of host species it can infect. We want to know if it comes from a part of the world where there's ongoing outbreaks. We want to be able to identify and track distinct lineages of virus. And crucially, we want to know if the virus belongs to any high-risk lineage like clade 2344 goose guangdong H5. So really the question becomes, can we actually use these tiny scraps of genome that we're fishing out of the mud to answer any kind of these practical questions? And fortunately, the answer is yes. So I'm going to show you 3 approaches we've come up with for interpreting these sequence fragments that we recover. So the first approach is that we compare the fragments to reference sequences from well-characterized viruses in global databases. So we queried the 19,000 fragments that we recovered against over 500,000 reference sequences that were annotated with their host species, their collection location, and their lineage/clade if it was applicable. And we obtained very strong alignments between our fragments and their best matching reference sequences. The median alignment identity was 99.1%, and the median alignment coverage was 100%. So this means that the fragments of virus that we were detecting were highly similar to viruses in the reference databases. So we could confidently infer things about these viruses from the annotations of their best matches. So for the next few slides, I'm just going to focus on H5 fragments because the goose guangdong lineage is public enemy number one. Now, in fall 2021, when the surveillance program began, the goose guangdong H5 viruses were only circulating in Europe and Asia. So the BC Ministry of Agriculture really wanted to detect incursions of these viruses into BC. Now, as it turned out, the goose guangdong H5 viruses didn't arrive in BC until last year in April 2020, or 2022 rather. So right in between our two surveillance seasons. And this was somewhat advantageous because it gave us an opportunity to compare a pre-arrival season to a post-arrival season. So in this heat map here, each row is one of the surveillance seasons, and then each column is a lineage/clade of H5 virus. And what you can see is that in the first season, the 2021-22 season, all of the H5 fragments had their best matches to local American non-goose guangdong H5 viruses. Now, the following season, in 2022-23, almost all of the H5 fragments were from goose guangdong clade 2344 viruses. Now, here's the same type of heat map, but this is for host species instead of clade. And what you can see is that in the first season, the H5 fragments that we recovered were most similar to viruses detected in waterfowl, especially mallards. And none of them were similar to viruses that had previously spilled over into poultry. Now, this really shifted in the second surveillance season after the arrival of the goose guangdong H5 in British Columbia. In the second season, the H5 fragments that we found in the mud were most similar to H5 viruses infecting poultry and some waterfowl. And there was also some similarity to H5 viruses detected in scavengers and raptors, which we know become infected when they eat other infected wild birds. And then one last heat map, same idea, except this time we're looking at the geographic range. So in the first season, all of the H5 fragments that we recovered were most similar to viruses collected in the US, so North American H5 viruses. In the second surveillance season, this shifted to H5 viruses from Europe and to a lesser extent, Asia and Africa, which is compatible with how the goose guangdong H5 virus migrated into North America, which was from Europe over the Atlantic Ocean via Iceland and Greenland. So altogether, these three heat maps tell a pretty clear story, which is that in the fall winter of 2021-2022, all of the H5 viruses circulating in BC birds were, I call them your garden variety North American lineages that are only really known to infect resident waterfowl. But the following year, in the fall winter of 2022-23, all of these local H5 viruses had been completely displaced by the arrival of this highly pathogenic clade 2344 goose guangdong H5 virus from Europe. So that's the first approach that we have for analyzing these genome fragments. And it really relies heavily on comparison and inference based on similarity to other viruses. So the second approach that we have, it provides a much more direct indication of the risk presented by the viruses we detect. So in this approach, we scan each of the fragments that we recover for specific mutations and specific genetic signatures associated with spillover phenotypes. And as an example, we're going to look at HA cleavage sites. So in order to enter the cell, the virus has to have its HA or hemagglutinin surface protein cleaved by a host protease. In low pathogenicity viruses, the cleavage site contains a single basic amino acid residue. And this motif is mostly cleaved by trypsin. So that really restricts replication of the virus to respiratory and intestinal epithelia. Now, in high pathogenicity strains, there's multiple basic amino acids inserted into this cleavage site. And this allows the cleavage to occur by ubiquitous furin-like proteases, which leads to these systemic infections that become really severe fulminant disease. So in the first surveillance season, we recovered 11 fragments of H5 that spanned this cleavage site. And all of them contained the very classic single basic low pathogenicity motif. Now, in the second season, after the arrival of the goose Guangdong H5 virus, we recovered 33 fragments of H5 that spanned the cleavage site. And all of them contained a multi-basic high pathogenicity cleavage site motif. And we actually observed 3 different variations of a multi-basic high pathogenicity cleavage site motif. So this cleavage site, it's just one example of a genetic marker we can look for. We can also look for receptor binding motifs. We can look for stabilizing mutations that allow the virus to adapt from avian body temperature and pH to mammal body temperature and pH. We can look for specific immune epitopes to see if there's cross protection. I mean, there's many other virulence factors and spillover factors that we can interrogate using this method. So it really makes it a pretty powerful tool for us. So that was the second approach. The third approach involves phylogenetics. Now, phylogenetics provides a really high resolution way to analyze viral lineages. And, we could use this to find out how many lineages within each subtype were present. We could use it to find out if the same viral lineage was present in multiple wetlands, or if a certain lineage persisted between surveillance seasons. I mean, there's a number of questions that phylogenetics can answer for us. The problem is that phylogenetic analysis requires complete genomes, and we're only getting these little short fragments. We also know that these specimens frequently contain multiple viruses, so we really don't want to try putting these fragments together because we don't know if they all come from the same virus. So really, what we had to do is kind of develop this new approach to phylogenetics that we're calling proxy phylogenetics. We're going to look at H3 viruses for this explanation because they provide a pretty interesting example in our data. So the first step of the proxy phylogenetic method is that we build a phylogenetic tree using full-length complete reference sequences from a global database. So the tree on this slide here, it represents 608 avian origin H3 sequences that were collected globally over the past 3 years. And clades of highly similar sequences have been collapsed down into single branches, single lineages on this tree. Now, the next step of the proxy phylogenetic analysis is that we take the fragments that we recovered and we align them to the reference sequences composing this tree to find their best matches. And the best matches for our fragments tell us which branches of this tree we detected. So on this slide, the lineages that we detected in the sediment are shaded black, and then the lineages we did not detect are shaded gray. And right away, you can see that we actually detected 7 different lineages of H3 virus in the sediment. Now we can take this a step further by using grid plots to further stratify the lineage detections. So in this figure, the lineage detections are further broken down by wetland and by surveillance season. So in the grid plot, there's a column for each wetland where H3 fragments were detected. The blue grid cells represent detections in the 2021-22 season, and then the green grid cells represent detections in the 2022-23 season. So this kind of analysis lets us see that there were 2 lineages that persisted over the summer of 2022 and were circulating in both surveillance seasons. And it also shows us that these lineages were widespread across multiple wetlands. Now, this analysis also shows us that there were actually multiple lineages of H3 co-circulating in the same wetland. So you can see there was a couple of wetlands where two different H3 lineages were co-circulating. And then there were a couple of wetlands where three different lineages of H3 were co-circulating. Now, some of you may be wondering what the size of the circles represents. So the diameter, it indicates the number of reference sequences from the global database inside each of these lineages. Now, since the global databases are mostly filled up by active and passive bird surveillance, the size of the circles probably shows us the surveillance bias of the global databases more than anything else. So the fact that some of our detections include small circles, like lineages with small circles, it's really telling us that sediment surveillance is probably detecting H3 lineages that are underrepresented by current surveillance methods. So those are more kind of like ecological questions that we can answer with this proxy phylogenetic, but we can also use this method to answer more practical surveillance questions like, are the viruses we detect in the mud the same ones causing the outbreaks on poultry farms? And that's what this figure gets at. So it shows H5 lineages detected in BC poultry versus H5 lineages detected by BC's passive surveillance program versus H5 lineages detected by our sediment surveillance. The green cell indicates lineages detected in poultry. The blue cells indicate lineages detected in sediment, and the orange cells detect lineages or indicate lineages detected in wild birds by passive surveillance. And then there's also beige shading for the goose guangdong lineage with the clades written next to them. So what this figure really shows us is that the clade 2344 lineage causing the outbreaks on farms in BC, well, it was the same lineage that was detected in wild birds by passive surveillance, which is something we also detected in the sediment. Now, what this figure also shows us is there's an additional lineage of H5 near the top that was only detected by sediment surveillance. Now, if we zoom in a little and increase the resolution of these lineages, the story becomes a bit more interesting. We can see that there's three sublineages that were detected in wild birds through passive surveillance. And we detected those same three sublineages in the sediment. But we also detected an additional three sublineages in the sediment that weren't picked up by passive surveillance. And another interesting point is that it was actually only one of these sublineages that ever caused an outbreak on a farm. So in this case, for both surveillance methods, they're suggesting that the range of threats that was out there was actually wider. Now, overall, so those were just H5s, but I repeated that same type of analysis for all the subtypes. In every subtype that we detected in wild birds by passive surveillance, we also detected in the sediment. And using proxy phylogenetics, we also confirmed that all the lineages detected by passive surveillance were also detected in sediment. And then our results also showed that there was numerous additional subtypes and numerous additional lineages that were detected in the sediment only. So basically, every flu virus we found in the birds, we also found in the mud. And then there was a whole bunch of other additional flu viruses that we found in the mud. Now, before anybody gets too carried away, we don't think sediment surveillance is a replacement for bird-based surveillance. Bird-based surveillance still has some advantages. First, you can confidently assemble a complete genome from a bird specimen. You don't need to worry about that problem of having fragments from different viruses and not knowing how to put them together. And as you saw, we rely on having a good source of complete genome sequences for our proxy phylogenetics and our comparison analysis. So I don't think we'd be able to do this if everybody gave up bird sequencing. The second advantage of animal-based surveillance is that animal specimens let us see which genome segments are present with each other. So unlike sediment, we can use animal specimens to detect and analyze reassortment events. And then finally, and probably most obviously, with an animal specimen, you can directly determine which host species the virus came out of. So the bottom line for us is that bird-based surveillance and environmental surveillance are really complementary methods, and deploying them together is likely going to be the most effective surveillance program. And that wraps up my talk for today. But before I take any questions, I just wanted to acknowledge that this avian flu sediment surveillance project is really a big team effort. We have Dr. Natalie Pristizecki at the BC CDC, who's leading the charge there, and Dr. Chelsea Hemsworth at the BC Ministry of Agriculture. And then there's Michelle Coombe, who's a PhD student at Ministry of Agriculture, and she's really the mastermind behind picking which wetlands we sample and when we sample them. There's Oliver Busby, he's the brave soul going out to these wetlands to collect the specimens. And given the time of year, it's like often not in the best weather. And then, of course, we have a dream team at the BC CDC of Sarah Mansour, Gab Cortez, Marzea Calor, and Ethan Kenyor, who organize and carry out the extractions and screening process at the BC CDC. And then also I wanted to thank Shannon Russell and James Vosnick at BC CDC because they provided some of the more recent unpublished sequences from passive bird surveillance. And then, of course, a thanks to the UBC Department of Pathology and Lab Medicine for hosting this rounds, and a thanks to all of you for spending your lunch hour listening to me talk about mud and bird flu. So with that, I'm happy to answer any questions you might have.
Great. I think we can open this up for questions. Are the same approaches being used for any other types of infections which cross species?
Yes. In fact, I had a ton of slides about that, but cut them out in the interest of time. So basically, this platform that we've developed, it's really specimen type and virus agnostic. So the tools that we built, they don't only work on mud and they don't only work on bird flu. So we've used them to do surveillance of novel coronaviruses and bats from the Congo. We're using them to do outbreak investigation and food supply chain monitoring for noroviruses. We're working on using them for wastewater surveillance of different pathogens. And there's a billion other applications you could dream up where this kind of technology would apply. Yeah.
Thanks. Are there any other?
There are a few questions in the chat, I think.
Oh, super. Well, Helen said not really a question, but she really enjoyed your slides, and I think we'd all agree they were great to look at. So for residual sediment samples, can you preserve them for future research uses or make a kind of a enviro biobank?
Yeah, definitely. So in theory, yeah, absolutely. I mean, the thing about the probe capture method that also sets it apart is that you build the library first. So you really just have a metagenomic library, and we capture bird flu out of them, but you could go back to that same library and try to capture something else, Salmonella or West Nile virus, or whatever. The only thing in practice that would make that a little suboptimal is the fact that we pre-screen everything for flu. So now, because of that, our biobank, as it were, is now heavily biased towards specimens that contain influenza A virus. But I mean, that being said, if you really did want to have kind of like an enviro biobank and spend a bit more money, you would just make a library out of every specimen you collected and not pre-screen them for a specific virus. And then you could keep going back to those same libraries with different panels of probes to capture different targets for different research questions.
Sure. And there's another longer question from Dr. Donahue. And as mentioned, Kevin, I have to drop off because I have to run to another round, which I'm hosting. And but I'll let you address Dr. Donahue's question. And there's another one from Dr. Cote as well in the in the chat. So thank you.
So Michael has a question about seasonality here. So except for bird migration, are there any other seasonalities to AIV prevalence in this population, the wild birds? So yes, there are. There's climactic ones. So during the summer, it's sunny, it's hot, and it's dry. So those are all three things that do not favor the survival of viruses or the transmission of viruses in an aquatic environment like a wetland. So that also partially explains why the fall winter is bird flu season, because then you don't have that hot sun irradiating the viruses or accelerating these degradatory processes in the environment. And then also during the winter, there's, I mean, especially here, there's a lot more precipitation. So the wetlands are wetter and cooler. So the just same way that you cryopreserve a specimen in a -80 or a -20, having cold temperatures, it preserves the environment and it facilitates the transmission of the virus in the environment. Another interesting one that's, I guess, kind of relatable maybe, is that there are also little mini surges of AIV activity around the times of the year when chicks are born. So just like human babies, avian babies also get their parents sick. And because you're introducing all of these immunologically naive individuals into the community all at the same time, they kind of incubate the virus. They're susceptible, they get exposed, they incubate the virus, they get the parents sick. So you do get a little blip of activity around that time too. So short answer, yes, there's more factors than just bird migration bringing new viruses in. There's also like the population dynamics and the climate as well. Are wild birds outside at all times, no matter what the season? I see, yeah. So that kind of ties into the environmental factors that affect the seasonality as well. Because, I mean, there are wild birds that use the built environment as urban wildlife. Like I'm sure we're all familiar with those geese who hang out outside VGH, right? So it does kind of affect the distribution and dynamics of the virus in the population, but I think it's such a huge question that nobody really has been able to study it and get a good answer as to how that affects it. Okay, Dr. Cote, do you think this approach could be used for CBS blood surveillance? Yes. I think in general, if we want to prevent spillovers and pandemics and even just do public health surveillance for pathogens, there's a lot of tools out there that we can use. Like there's a lot of sources of specimens, creative sources of specimens that we could start using for genomic surveillance. And I think blood banks, they're an excellent one. I mean, they're already used for screening a very specific list of pathogens that are like bloodborne. But I think you could use that as a source of specimens to do genomic surveillance for other pathogens. Now, the only problem here is you're kind of really it's only going to work on viral pathogens that cause viremia, because otherwise you're not going to have viruses in the specimen. But that being said,
some pathogen would be in the blood cells.
possibly, yeah, possibly. But I mean, like influenza, it's like it's mostly epithelial replication. But that being said, I think there's other ways to look for evidence of infection than genomics, like high dimensional serological assays. And in that case, I think the blood banks as a source of sera would be fantastic for doing those kind of seroprevalence screens.
All right. Thanks.
Can I ask a
Tyler.
question? Yeah. So Kevin, really good talk and super interesting, and it's cool that you guys are able to do this with these samples. So in a perfect world, if everyone's doing bird surveillance internationally, how might you use that data to try to limit trends like the virus jumping to humans?
Yeah, that's a really good point. Because I mean, I know I tried to disguise it as an advantage that the bird flu came between our two surveillance seasons, but really what it meant is that our entire program missed the incursion that it was trying to detect, which was unfortunate timing, really. And the reason that like it was because it came in through the back door, so to speak. Like it came in to North America through Newfoundland.
Yeah.
And then because normally historically we're accustomed to the Pacific Flyway bringing the bird flu into British Columbia and the Pacific Northwest. And so our entire surveillance strategy and the timing that we chose is based on that migration.
Yeah.
The argument becomes like, well, if we had partners in Newfoundland and all of these other flyway choke points, right? And just have like an integrated North American surveillance system, then it would probably function much better, right?
Right. Yeah. And so then would the idea be that if you had an area, because the issue is it's spreading internationally through the birds anyways. So is the question just about trying to recognize those potentially human pathogenic, like, because like it's still hard to see how you could
what do you do about it?
Yeah.
Yeah, so that's a really good question. And basically what it comes down to is biosecurity on farms. So there's actually like biosecurity requirements that ministries can impose on producers. And generally they're a pain in the neck and they're costly. So nobody wants to do them. And they can be like as simple as you need to change footwear upon entering a barn if you're coming from the outside, to like you are not allowed to drive your vehicle off your property for the next 3 months, which, as you can appreciate, makes it extremely hard to do business, right? So the producers also don't like having the government come in and call their entire flock. So they are amenable to increasing biosecurity, but when the evidence is there for it. So that's really what it comes down to.
Yeah, so you could have more targeted interventions for specific bird species in specific geographic locations.
Yeah, and I think this is kind of a lesson from COVID. I know nobody wants to talk about COVID anymore, but it's that once the spillover into humans happens and that final stage of adaptation has occurred, the cat's out of the bag. And whatever control measures you use are going to be reactive. You're not going to stop a pandemic that's already started. So that's why
it's easier to contain birds than it is humans.
Well, it's easier to focus on that interface of interaction between wildlife and poultry, and then or livestock, and then livestock and humans, and have the interventions there, rather than wait for the pandemic to start and then try to chase after it.
Yeah, that makes sense. Yeah.
Yeah.
Cool. Thanks.
Mm-hmm. If anybody has any other questions, I think there's like an option to raise your hand here somewhere.
There is, but we're
Maybe
at
I'm
12.
thinking of Microsoft Teams now.
I think we're at 12:57. I think we could probably wrap up.
Great. Well, everyone.
Yeah, thanks, Kevin, for the really good talk. And I think next week we're going to have a PAL session, but on quality from Mike Noble's successor as our UBC quality lead. So look forward to that next week. Thanks everyone for tuning in.
Thank you, Kevin.
Thanks, Kevin.
Thank you all. See you next week.